Good morning, everyone. So it's a pleasure for me to share with you the recent acquisition that we had in my laboratory. That is the laboratory of the local healthcare center for Romagna in Italy. So my talk of today will be dedicated to the point of view of the microbiology and in particular virology laboratory for the management of the current COVID-19 pandemic. I'm just showing you some cumulative integrated data from the surveillance we have in place from COVID-19, and these data are just until July. That means at the end of the first wave, and we are now I would say almost in the peak of the second wave, but in July, in mid-July, the data were just getting better and better day after day, because we started to put a lot of pressure on the RT-PCR, on nasopharyngeal swab, because since the local healthcare system in Italy is regionally based, we need to go, and any region needed to go for the implementation of a complete system for RT-PCR testing. A few words to describe what is the present healthcare organization of my region. That is the one you see here in red. We had three major areas. One is the so-called Area Vasta Emilia Nord, here in pink. The second area is the Area Vasta Emilia Center. That is the one you see here in almost green. And the third one is the area in which my laboratory is located, that is here depicted in colored in blue or light blue. It is the area of Romagna. Any of these areas as subsequent local health organization. So that means that we have the territory of the whole region, I would say, in quite a strict control, because there is a strong integration in between the hospital located in each single area and the healthcare services that take care of the local territory. That means we have a model in which the integration in between the hospital care and the territorial care is quite strictly integrated. Probably not as much as it would have been necessary for a perfect control of this COVID-19 epidemic, but we still believe that our model of integration is quite good. So this is the daily number of COVID-19 swab performed by the different region, and this is the ratio of swab tested per 100,000 inhabitants per area. And not getting into much detail, you see these 20 bar represent the 20 region of Italy, and you see that the ratio of sample tested per 100,000 inhabitants is much different from one area to another. And the average value of testing for 100,000 inhabitants at mid-July was about 82, of which 40% was dedicated to control swabs, and almost 60% was dedicated to swabs for the diagnostic of suspected cases. This is the average in the nation, so we had about 82. But in our area, even at in the middle of July, we were testing more than double the ratio of the nationwide system. So we were testing already a lot of swabs per day in order to keep the system under strict control as much as we could. And you see here the different province of my region, the Emilia-Romagna, and you see that the ratio, the trend of contagion is not equal. So we started with differently affected area, and this is probably quite easily explainable according to the concentration of population in the different city. Bologna is the capital city of the region, and so it's probably one of the most crowded. And even the area you see here from Reggio Emilia to Parma, including Piacenza and Modena, are very crowded on the territory. So that means that these central and northern part of the area have been more affected because of the increased population in a smaller territory. And another issue that we need to discuss is the mortality rate that we had in Italy. And we had one of the highest mortality ratio according to the report I saw very recently in the literature. And on average, we had in the first wave 14.3% mortality, with a peak in Lombardy that was more than 70%, and the lower level in a smaller region like Umbria and Molise, where the mortality ratio was about ranging in about 5%. So again, we have a lot of differences. And I need to provide you some additional hint on my laboratory organization. We serve all the area here in the southern part of Emilia-Romagna, in Romagna actually, and the laboratory has an hub and spoke configuration, and we have 7 urgency laboratories that are located in the 7 major hospitals that are open 24 hours a day for 7 days in a week, and 1 central hub laboratory located in the center of the area here. It's quite an unpleasant place, I would say. We are in the middle of nowhere, but it's the right place to build the laboratory because it's here, exactly in the interconnection in between the highway that connect west to east and north to south. So we are in the perfect center. We have 3 different units in this hub laboratory. One is clinical pathology. The second is my unit of microbiology and medical genetics, and we do some 21 million tests per year. And of course, we need to collect the sample all over this area. And this is more or less the situation of our traveling system, our logistical system, from the peripheral sampling point that are more than 100, and the major hospital, all the trucks that transport the sample to Pieve Sistina, that is the location of my laboratory, are traveling up and down on the highway, and sometimes this highway are really very crowded, because you see here, this is a picture taken probably in the middle of August, when most of the Italians and also people coming from abroad are just traveling on this highway to get the shore of the Adriatic Sea. That is one of the most popular spots for tourism in Italy. So, given this situation, I would like to stress that we do need to have available a collection kit that allow us to manage all the sample in the right way. I'm sorry, I'm losing my screen. Oh, I'm not getting my screen anymore. Okay, so I'm sorry. Yeah, we need to have this kind of transport medium that can allow us to ensure that the sample are correctly stored over the logistic transportation. So we selected the UTM, Universal Transport Medium, that contain protein that allow the virions to be stabilized, and also some antibiotic and antimycotic that, of course, are very useful to block the overgrowth of bacteria and yeast that are for sure contained in the nasopharyngeal and the oropharyngeal secretion. These medium can also guarantee a kind of cellular integrity and the preservation of the vitality of viruses. So the tube also contains glass beads that allow homogenization of the specimen, and the tube could be stored at 225 degrees for up to 48 hours. That in this kind of logistical situation, it's very, very, very useful. And by using this collection device, you can just perform as much test as you want, because the average content of the tube we are using is 3 milliliters, so we have plenty of sample, and you can go for cell culture, direct immunofluorescence, lateral flow, rapid antigen, assay, PCR, direct fluorescence of IFA, and it's really plenty of possibility for the use of UTM downstream the collection of the nasopharyngeal or oropharyngeal secretion. And of course, the FDA is, as well the ECDC and the WHO are strongly suggesting that the collection of nasopharyngeal secretion by using a nasopharyngeal swab is the best sample possible for detecting SARS-CoV-2. Of course, as an alternative, also a oropharyngeal specimen can be collected, and the nasal mid-turbinate could also be collected. But anyway, the nasopharyngeal collection is probably the best one. And of course, you need to ensure that the nasopharyngeal secretion are immediately placed in a secure and safe transport medium in order not to lose any RNA or any antigen you may need to detect for diagnostic workflow. So it looks like that collecting the nasopharyngeal secretion is a simple procedure, and actually it is, but you must be very careful, because when collecting the sample, you should bear in mind that the nasopharyngeal space is not here, is just down in the nasal cavity, in the middle of the space in between the eye and the hair. So you need to get very deep in the cavity, and you need to rotate at least for 10-15 seconds the swab in order to collect a good sample. After that, you can just take out the swab from the cavity and place it immediately in the red capped tube, in which you can just put the lower part of the swab, because there is a breaking point that is very convenient. Of course, we have 15 hospital and outpatient, and you see that the sample collection is taken in the peripheral area. Well, this is not actually the right number. This number was related to July, when the first wave of the epidemic was just almost down. Now we are collecting something that is almost 3 times 2,500. The average is about 6,900 per day. But anyway, we try to manage. Of course, the swabs are transferred in the UTM to release the virus content into the medium, immediately transported to the laboratory, and once the laboratory receives the sample, we start to prepare the sample, and we go for net processing. That is basically RNA extraction, RNA rate to transcription, cDNA amplification and detection, but we are using machines that are performing these 4 steps only in one time, and then we go for clinical classification. In general, we are capable to provide results for the most urgent sample that are coded as red sample in 2 hours from the arrival in the laboratory, the yellow that are intermediate urgent test in 6 hours, and the so-called green sample require about 24 hours for being completely reported to the clinical site. But this is particularly relevant because we need to classify the sample in a good way in order not to, not to face the true requirement that we get from the pandemic control officer and the doctor in the hospitals. You see here the number of cases. The new outbreak started probably end of July, you see here, and the number now are more or less the same we had in the first wave, probably a little bit higher in terms of ratio of positivity, because we are not only controlling the suspected cases, but we are also controlling all the suspected contact cases. That means we need to test many more people in order, and of course the detection rate is higher. The SARS coronavirus 2 is just the third of a series of coronavirus that jumped from the animal into the human beings, thanks to their capacity to vary their genome and to make them capable to infect humans. The first one was the Middle East respiratory syndrome in 2000. Well, it was the SARS-CoV-2, sorry, the SARS-CoV-1 in 2003, starting in Hong Kong and in southern of China, and then followed by the MERS-CoV Middle East respiratory syndrome coronavirus in 2012, and then we have our SARS-CoV-2 2019. And we have many different, many reference sequence for SARS coronavirus 2, and fortunately, we could say that the coronavirus 2 genome is quite conserved, especially in the E6-7B region, where no mutation was so far reported. And we have hotspot for mutation in ARF1A, S8, and the N region. And of course, this mutation can lead to two major problems. The first one is a variability in the possibility to detect the viral genome if you target your test on these hotspot for mutation. And in the case that is not so frequent, but it's possible indeed, so you must consider also this possibility. In the case this variation in the genome will generate variation in the amino acid sequence. There could be some relation between these genetic variation and different capability to infect human beings. So by detecting different components of the virus, you can achieve the diagnostic. And you see here, you can detect the RNA by using different techniques, like the PCR amplification, NGS, or isothermal amplification like LAMP or TMA, and you can also go for more sophisticated technique like CRISP. But on the other hand, you can just detect the antigen by using ELISA or lateral flow assay and by using chemilumination technique. So you have two different possibilities to detect the different components. One is the genomic-based technique, and the other one is the detection of different antigenic components that are emerging since the last, I would say, 4 to 6 weeks on the market as alternative method for the detection of SARS coronavirus 2. And of course, you have also the possibility to indirectly detect the infection by identify the antibody response that the infection is eliciting in the infected people by using, again, ELISA or lateral flow assay. And the reference method is indeed the so-called serum neutralization assay, SBN, or plaque reduction neutralization test, PRNT. Of course, we have many different possible weakness points in the analytical diagnostic workflow, but we need to focus also on the pre-analytical steps, because the pre-analytical steps are influencing in a very relevant way the result of our analytical procedure. I want to focus your attention on the statement in red, because the statement in red are probably more related to, more capable to influence the results of the testing that we will do on these sample. The first one is an inadequate procedure for specimen collection, handling, transport, and storage. I believe that the most relevant point is to collect the sample, the nasopharyngeal sample, by using a correct procedure. I told you before that a good sample is taken if you go into the nasopharyngeal space. That is not the nostril and is not a superficial space, is a very deep space. You need to get there, and you need, second statement, to collect the appropriate material for quantity or volume. That means you can not just get into the right space and take out the swab, but you need to stay there for at least 10 to 15 seconds. Of course, we have also analytical issue that needs to be addressed. Of course, we will see later in this presentation that in the testing procedure for any virus, but for COVID-19 related virus is even more relevant. You need to test the patient inside the right time frame. If you test the patient too early or too late, you will get I would say an analytical good result, but is a false negative result under the clinical point of view, because the patient is correctly infected, but you cannot detect the RNA if you test the patient too early or too late in the course of the disease. And of course, another issue is that, but this is quite obvious, that you need to test only validated assay that are at least for EU, CE, and IVD mark. You cannot test this sample by using not certified test. Of course, the gold standard for the etiological diagnosis of SARS-CoV-2 is still today the reverse transcription real-time PCR that must be performed on upper and lower respiratory tract specimen. The RNA isolation is one of the crucial points that I would say nowadays is quite unnecessary. It really depends on the selection of the test you are using, but there are many tests available on the market that are not requiring a specific RNA extraction. And this is very relevant because if you go for this kind of test, you can save a lot of time. So that means that the productivity of each single laboratory is largely increased by selecting these samples that are not requiring the pre-step of RNA extraction. Not all the tests are working in that way, but well, I know that on the market there are some tests that are performing in this way. Of course, the RNA is amplified, and the best results so far are obtained with the combination of different target genes. A good test for SARS-CoV-2 should not be targeted to less than 2 different genes. And the combination of the most widely used gene include the RDRP, that's the RNA-dependent RNA polymerase, and the genes E, N, and S. In general, the testing sensitivity is about 95%, and the limit of detection is lower than 10 copies per reaction. Now, I would like to spend some few words on this extremely low limit of detection. A major concern is raised since the starting of this pandemic situation about the true clinical meaning of reporting extremely low viral load in sample from patients that have been cured or they are completely asymptomatic. In other words, I'm not sure if a very low viral load, maybe lower than 10 copies per milliliter per reaction, is really relevant under the clinical and epidemiological control of the situation. It's really likely that these very low level of RNA that are detected by these extremely sensitive methods are not capable to infect anyone. We tried to isolate the virus from some almost 100 right now sample showing this extremely low level of viral load, and apparently no virus can be grown in cells according to this very low viral load. So we have been using also the serological assay for a while, and I must confess that nowadays the serological assay are probably of lower level of interest because they have a limited sensitivity at an early stage, and the average time for seroconversion is in between 10 to 14 days. They have a not completely defined level of cross-reactivity between antibody directed against coronavirus and other coronavirus. Of course, the sensitivity and specificity of the serological assay are affected by the target antigen that are including mainly the S1 and S2 protein and the N protein as a target in general. And of course, is well known the lateral flow assay as, in general, a lower sensitivity than more sophisticated tests like the enzyme, the ELISA, and the chemiluminescent assay. So I don't want to spend too much time on that. Okay, so we are getting close to our conclusion, and this slide is very interesting because this summarizes the possibility to apply different methodology to the diagnostic of SARS-CoV-2 infection. Of course, if you look at the graph here that report the viral load in the upper respiratory tract secretion, you see that the infection starts here. And of course, about 5 days after the infection, we have the onset of symptoms, and the viral load starts to grow very fast in the next 5 to 7 days, and then starts to decrease. And in general, in between 11 to 15 days, the symptom relieves and the patient is clinically resolved. And I would say that you can only apply RT-PCR in these diagnostic windows, shaded in light gray. If you apply for RT-PCR earlier than 5 days before the onset of symptoms and later than 15 days, you will probably get false negative. If we apply the same picture to the antigenic test, I mean the tests that are capable to detect the antigen deriving from the virus in the same material, in the same upper respiratory tract infection, these gray shaded areas should be moved in this direction. That means later in the course of the infection, because the detectability of the infection by detecting the antigen is probably starting someday after the onset of symptoms. It could last a little bit more than PCR for the symptom relief. Again, this is a very simple scheme that could be useful for the interpretation of the results. I don't want to take too much attention and spend too much time on that. And just trying to provide a high level of service, we need to consider all the statements in this slide in order to minimize the risk of diagnostic error for the correct identification of SARS-CoV-2 infection. So we need to have clinical and epidemiological evidence. We need to have chest computerized tomography that provide us with a lot of information about the actual status of the of the lung of the suspected patient. We need to collect in the right way the sample for RT-PCR or for antigen detection. We need to provide clear instruction about the collection procedure of each swab we can collect. And serology could be used as a confirmatory test in the patient with negative PCR after 45 days. So this is probably one of the most relevant features of serology. It holds a strong and very relevant potential to be used as confirmatory test in all of the patients that are clinically and epidemiologically defined as COVID-19 affected patient, but they do not receive a confirmation by RT-PCR. And at least in my experience, this percentage of patients that are clearly COVID-19 patients, but they didn't get any confirmation by PCR, it's about 20%. So that's quite not a completely relevant, but not irrelevant the same way percentage of patient. Okay, just to summarize again, the COVID-19 patient can be tested by rapid test and by concentration of oxygen in the blood. The patient can be tested by nasopharyngeal swabs, by RT-PCR molecular test, or antigenic test. Of course, if the patient goes to the hospital, the CT scan and the ultrasound evaluation must be performed. Also, the blood can be taken for confirmatory tests we just discussed. And I believe that that's it for today. Thank you for your attention. And of course, I will be more than happy to answer any question you may have by sending me an email, and I can provide you any clarification. Thank you.